welcome everybody to the AgResearch Pesticide Resistance Management webinar. Sarah's introduced me sort of, and I'm Paul Munro. I'm one of the technical leads in AgResearch, and I'm beaming in to you from Taupo. Part of my role as my technical lead role has been to facilitate the updating of the pesticide resistance management strategies, which are on the New Zealand Plant Protection Society website. And that's led us to this point in time for this webinar. We have about, I think about 120 people that are online at the moment. And we do have 2 hours set aside, but I'm pretty confident that we probably don't need all of that time. So what I need to do now is to move into my slides, and I'm unable to request all right. Righty, now I think we've got some action here now. And so the purpose of this webinar is to, I suppose, advise that there have been some changes over the last 3 to 4 years in the pesticide resistance management strategies that are on the website. And we're hoping that with some of the presentations that we have today, that there'll be some ideas from the people that are online who may see that there could be some gaps in the strategies and provide us with some guidance. And if there are any, it would be useful to know which ones are more important than others. But I'll lead on to that a bit more later on. The agenda today is we've got 3 expert panelists. We've got Trevor James, who's a lead scientist from AgResearch, Rob Beresford, who's principal scientist at Plant and Food Research, and Tim Herman, who's the senior tech services specialist at BASF. But what I'd like to do to start with is perhaps provide a bit of history of how we've got to this point. So the New Zealand Plant Protection Society established the New Zealand Committee on Pesticide Resistance back in 1987. And the task that group had 3 task groups for each of the fungicide, insecticide, and herbicide portfolios. And those groups all had a leader and were made up of other personnel who were either registrants or industry experts, and we had some scientific oversight, and the last person that provided that scientific oversight was Rob Beresford. And you've got to bear in mind that this work was all done by volunteers, so no one was paid to do any of this. And the purpose of these groups was to develop local guidelines, strategies that are based on the overseas information from FRAC and IRAC and HRAC. So there are obviously going to be some subtle differences between what happens in New Zealand and what happens overseas, but the guidelines, international guidelines, will always be the base. And these guidelines were all hosted on the Plant Protection website and were available for anybody to view. But probably more importantly, that the pesticide labels where they required a resistance management strategy all pointed towards the website to go and explore to find more information that wasn't on the label. And there has been limited activity over the last 5 or so years because everybody has a day job, and just things hadn't been kept up to date as we would have liked. Another part of the history is that couple of 3 years ago, 2019, in response to a formal request by the committee to AgCarm. They requested that the AgCarm become more involved in the management coordination of the resistance management strategies, and that proposal was considered by the AgCarm board back then. And the main changes that were proposed was that it would involve AFANS taking over the responsibility of the secretariat activities from the Plant Protection Society, and it would involve AFANS members becoming more directly involved in the resistance management strategies and develop the technical oversight for those. A Lighter Touch started in 2020, and we had funding to update the strategies. Part of my work plan was to update these strategies over the 6 years of the program, which were 6 years that was left. So we started off with a workshop in April 2021, where we had industry people, and some of those people are obviously on this webinar today. And the purpose of that was to look at what was currently on the website and what we needed to do to fix it. And we discovered that we had mode of action tables, and 25 of the strategies were significantly out of date and needed urgent attention. So we then thought, if we leave some of these to year 6, there are going to be some of these strategies are going to be more than 10 years out of date. So we decided that we would actually do them all at once. And so we contracted updates for the insecticide and the fungicide portfolios, and that was completed within a year. And we contracted Mark Christie, who was ex-DuPont and FMC, to go through those, which were downloaded from the website, and we went through that exercise. Luckily for us, the herbicide portfolio was pretty current because of an NB-funded project looking at glyphosate resistance and other weed resistance that Trevor James and Kerry Harrington were involved in. And the next steps were that the updates and the changes that Mark had made. We've had Rob Beresford peer review the fungicide portfolio, and Tim Herman has been looking at the insecticide portfolio, which is why those two gentlemen are on your screen right now. And we've started uploading the new strategies. The fungicide strategies have been uploaded already, and the insecticide ones are in progress. For the future, after this webinar, we hope to run a further workshop, which will be a collaboration, I hope, with A Lighter Touch and AFANS like the last one. And the purpose of that is to further identify the gaps that there are in strategies that need to be initiated, so written. We're looking to identify who could be contracted to do that. And what I mean by contracted, they would be paid. And we need to re-establish the 3 task groups. And some of that may be voluntary, but some of that might not be. And re-establish also the scientific and technical oversight to make sure that everything that is presented is accurate. And obviously this is all going to come at a cost, and we probably need to identify some sort of funding mechanism to maintain these strategies and the website as well. In addition to that, in A Lighter Touch, we've recognised that our growers are probably not familiar with the strategies that are on the website. They probably do not visit there. But given that these are the people that actually use these products in the field, we are going to develop an extension plan to provide these people with some user-friendly summaries on resistance management strategies, so that they can understand and obviously aid to the stewardship of these products. So that is all I was going to say at this point, and I've introduced our 3 panelists, and the first person who's going to be talking to us will be Trevor. Thanks, Trevor.
So you can hear me, I presume, and I'm just requesting control of my slideshow, which hopefully will come up soon. But by way of a bio for myself, I am based with AgResearch at Ruakura Research Station, Hamilton, New Zealand. And today I celebrate 50 years in the job with AgResearch and its predecessors. So I've been working with weeds for 50 years. I think that might qualify me to give the presentation which I'm about to do. So we will try and move right along, but I can't see any control for the slideshow. So here we go. Okay, so I will start this presentation with some definitions just to make sure that I will be hopefully understood a bit later on, and then a brief history of herbicide resistance in New Zealand, and then why are we revisiting herbicide resistance strategies and the next steps. So I'll start with target site resistance, because we're talking about two different ways that mutations cause resistance through evolution. And target site resistance is herbicides generally work by inhibiting an enzyme that produces a critical amino acid. And target site resistance inhibits the herbicide by changing the structure of the target protein that decreases herbicide binding at the site of action. And you'll see that in the diagrams to the right. And it could also actually, the mutation, cause a larger number of those genes, meaning that the herbicide is insufficient to block them all. So again, the plant will continue growing. If we look at non-target site resistance, this can involve multiple genes, and it works by decreasing translocation of a herbicide to its site of action. And you'll see on the right-hand side, the arrows pointing to where it might go, or it could increase metabolic detoxification of the herbicide, and or sequestration or immobilization of a herbicide in a part of the plant so it cannot reach the site of action. Now, this diagram here is actually about the resistance of glyphosate that can work through any of those different ways. So, and as well for glyphosate, there is one more method of resistance, which is also the target single site mutation. So we have some herbicides that use target site as their avenue to resistance. We have some that use non-target site, and we have some that can use both depending on the plant. So if that's confusing, we'll try and sort it out a bit later on. Talking now about the Herbicide Resistance Action Committee, you'll hear about this one where HRAC, you'll hear about FRAC and a few others later on in the committee. But this committee grouped the herbicides according to how they kill the plant. That is their mode of action, or MOA. For many years, different countries developed their own labeling system for MOA groups, and New Zealand was among those. And if you know your history, you'll know that we used alphabetical letters. We had Group A, Group B, Group C, etc. In 2001, it was decided to ditch New Zealand's unique system and run with a new HRAC system that was being accepted by all countries. So it's globally accepted now. This new system is based on numbers for each mode of action group, which is good because that clearly differentiates it from our old system. You can find more information about this on the New Zealand Plant Protection website, which is given below. But in a brief summary, there are 1 to 34 numbers for HRAC groups, and then they added a group 0 for where they don't understand the way the herbicide works on the plant. And for example, group 1 is the fops and dims, those grass killers, haloxyfop and sethoxydim, etc. And group 2 is sulfonylureas, which is metsulfuron, chlorsulfuron, those ones, and we go on and on. But not all groups have representative herbicides in New Zealand. So when you look at a group table for New Zealand, not all numbers will be there because we don't have those herbicides. And for either specific groups or specific herbicides, we have currently 7 strategies produced. And these are there to assist in the curbing of evolution of herbicide resistance. These were mostly written by Kerry Harrington, who has now retired. And again, more information on those specific groups is at the web link below, but it might be easy just to go in through the upper level New Zealand Plant Protection Society site, which is the shorter one below, and there you will clearly find your way to the website of herbicide resistance. So just now I want to talk about cross resistance. This is when a plant develops resistance to a particular mode of action and then automatically becomes resistant to other herbicides in that group. Now, this might be every herbicide in the group, or it might be just a selected few of the herbicides in that group. This also adds another layer of complexity to herbicide resistance. On the other hand, multiple resistance is when a plant develops resistance to 2 or more mode of action groups. And you will see this a little bit later on, examples of this when I talk about some specific herbicides. So how bad is herbicide resistance in New Zealand? So it was first discovered in 1979. And if you do your maths, and remember I've been here 50 years, I was part and parcel of finding this first case of herbicide resistance. The growth of resistance identified grew steadily. That's the graph there on the left-hand side. Grew steadily through to 2018. And then, as you've heard from Paul, we received 5 years of funding from MBIE to focus specifically on herbicide resistance. A lot of that work was looking for new cases of resistance via herbicide surveys. And there we had the cooperation of Foundation for Arable Research and Brigado Research Institute to help with those surveys in arable cropping areas and vineyards. As a result of that, we found 13 new cases of herbicide resistance in the last 5 years. So this slide tells us what those cases were, and I would just like to talk to them briefly because they show the breadth of herbicide resistance, the fact that it's not isolated to any particular crop, any particular industry even, or not even necessarily to agriculture. So starting at the top left, annual POA, this was found to be resistant to haloxyfop, a group 1 herbicide, which is why the number in brackets refers to the group. A group 1 herbicide, and this was found in turf on a golf course. It was also found to be resistant to iodosulfuron in cereals. So here we have a case of multiple herbicide resistance. The next one, sow thistle, was found in cereals, as well as willow weed and rayless chamomile. So they all came out of the survey of arable fields. Going across the next row, we have, there was one there right on the top right there, ryegrass was also found in cereals, as were prairie grass and lesser canary grass on the next row. The wild carrot in the middle there was resistant to glyphosate, which was a new one. This was the first find of glyphosate resistance of wild carrot anywhere in the world, and that was found in a vineyard. Then we have Spurry. This is interesting because this one became resistant and was found in clean crop brassica, where chlorsulfuron can be used because it's tolerated by the clean crop brassicas. And so we have Spurry there resistant to group 2. Down the bottom, we have summergrass, again resistant to a group 2 herbicide, nicosulfuron. This was found in maize. And then we have 2 more, prickly sow thistle and ripgut broom, which were found in the cereal survey. And then the last one there, blackgrass. Now you're saying blackgrass isn't in New Zealand. You are quite right to a certain extent. So this was found in as an incursion weed. It was actually found while we were doing the survey work in cereal crop, but it actually was an impurity in linseed that was imported from France and got sown accidentally. And when we looked at that, we found that it was already herbicide resistant. Okay, so which is another interesting thing. If we get impurities that come in and get accidentally planted with seed in New Zealand, they might already be resistant to herbicides. So can we predict what's next with some accuracy? This is a messy slide. All I want you to concentrate on is the stars. So here we predicted weeds that might become resistant to herbicides in maize and in cereals. And focusing on the stars, you will see that of all the resistant weeds in New Zealand in maize and in cereals, we have managed to predict each one of them. So prediction has a definite place. We can identify with a high level of certainty what the new resistant weeds might be. And this could be critical in writing strategies. So prediction can be helpful. So for crop-based strategies, our prediction can advise on species with a higher risk of herbicide resistance evolution in specific crops, which means that we can highlight that in the strategy and growers can keep an eye out for them. And the first time they see those things causing a little bit of trouble, not being well controlled, bingo, we can hit the issue early. Similarly, for herbicide or mode of action based strategies, we can predict which species have a higher risk of evolution of herbicide resistance from that herbicide, an individual herbicide like glyphosate, or from a whole mode of action. So once again, being able to put this in front of growers of what the problem might be. And specific management options then could be developed for these specific weeds. If you see it, let's target it. This could be too much for a strategy, but it's something that should be considered and we might like to look at. So more curveballs. So I went to the Australasian Weeds Conference last week in Australia, in Brisbane, lovely place. We won't talk about that. Paul Neve was a keynote speaker. We've been involved with Paul Neve for herbicide resistance for ages. He led all the resistance on blackgrass in the UK, and so has an extensive knowledge of herbicide resistance. And up until now, this is background, up until now, mixing herbicides with different modes of action was seen as a good tactic for delaying the evolution of herbicide resistance. So you go in there with 2 herbicides that control the same weed. We were saying, yes, they've got to evolve resistance simultaneously, otherwise they are going to be knocked out by the other mode of action, and therefore only in that rare case would we get herbicide resistance, which would be 1,000,000 times 1,000,000 or 10 million times 1,000,000. We were looking forward that to be an answer. And indeed, the work that Paul Neve found in the UK was that mixing herbicide modes of action did delay the evolution of target site resistance. So we're coming back to those definitions now. It did delay the evolution of target site resistance, but he also found that using mixtures of herbicides actually promoted the evolution of non-target site resistance. The complexity of this is too much to go into in this presentation, but you'll have to take it from me that that is a reality which we might have to consider. So the outcomes, we will need to rethink and rethink very hard many of our herbicide resistance strategies to how we might deal with mixtures and recommendations that on the one hand might delay target site resistance, but on the other hand might help the evolution of non-target site resistance. So just quickly looking at some groups there, only a few herbicides actually evolve based on only target site resistance. And 3 examples here are quizalofop, or 4 actually, from group 1, aryzal and trifluralin from group 3, picloram from group 4. A greater number of herbicides evolve both target site and non-target site. We've already heard about glyphosate, but group 1 herbicides tend to use either mechanism for resistance. Group 2 as well, dicamba and 2,4-D in group 4, and atrazine in group 5, as well as glyphosate. And then we have a greater list again of herbicides that evolve solely through non-target site based evolution. Bromoxyl and bentazone from group 6, glufosinate group 10, clomazone from group 13, alachlor, metolachlor and flufenicet from group 15, paraquat from group 22, mesotrione and topramezone group 27, and amitrole from group 34. So you can see how complex I'm creating the situation. So what does this mean? This is my last slide. So second last slide. Problems for New Zealand resistance management that would need to be considered in the strategy. We have a limited number of active ingredients. You compare to Australia or America, we have less than half the number of active ingredients that we can include in any rotations and other options that we choose for halting or stopping the spread of development of herbicide resistance. And these are often concentrated in 1 or 2 groups. So if you look at herbicides for control of grasses in cereals, are mostly in group 1 or group 2. And we have also few non-chemical interventions available. So there just isn't out there. These are being developed, and there's some great ones that are in the pipeline, but are still a little bit too expensive for many growers in New Zealand, which tend to be on a smaller scale than many of the others in the world. And also many growers are not set up for pre-emergence soil-incorporated herbicides, which is another good strategy for delaying the evolution of herbicides. You know, so that's in perennial crops or a no-till cropping, arable cropping. And then we have the problem of the resistant allele in the soil weed seed bank. So if we have a resistant plant and it sets seed, that seed goes into the soil weed seed bank. It can persist there for many, many, many years. The old cliche, 1 year seeding, 7 years weeding is more like, you know, up to 1 year seeding, up to 70 years weeding. So that is also a problem, is that this resistant allele can repop up in a plant at any time in the future. And also then there is the movement of resistant alleles from outside your boundary, either as mobile seed blowing in on the wind or with pollen, which is horribly mobile as well. And this can be from neighbors, from roadsides, or it can come in on machinery, etc., if our biosecurity, on-farm biosecurity, isn't up to scratch. So where to from here? In conclusion, clearly the weed herbicide strategies do require updating. The last resistant weed was added in 2021. We've got half a dozen more to add since then. We have much more information we can add based on prediction and that sort of thing, so we can be more directive in the strategies if that is what people want. But also there is confusion around herbicide mixtures and in the strategy, how they use. We need to resolve that through many heads getting together and talking about how that might be resolved. And then we possibly, well, I'd say probably actually need to add some new crop or mode of action strategies as well in the light of the latest cases of resistance and what we've found through the 5-year program that we had on herbicide resistance. And I believe that's it. And if I push enter again, it's all gone anyway. So thank you, Paul.
Can you hear me all right?
Yep.
Right. We have got some questions here, Trevor. The first one, I'm assuming, relates to herbicides, but is the rate of resistance development in New Zealand similar to other countries?
I don't know how to answer that one because I'm not sure where the question's coming from. So I'll give two different options. Within a plant species, within a weed, the rate is constant everywhere. So if we get a mutation rate of 1 in 100,000 or 1 in a million, that's irrespective of countries. The evolution of resistance in the actual industry in New Zealand has tended to be a bit slower than other countries. That's either because we don't have a weed that has a high predisposition to it, like rigid ryegrass in Australia, or our practices have already contained methods of decreasing it. In arable, this has been the pastoral phase, which for many years farmers looked at as an important part of their rotation. With intensification of agriculture, some farmers are dropping out this pastoral phase, and that's going to increase the speed or the rate of evolution if we're not careful. Do you think that's answered the question?
It has for me.
All things equal, it should be the same as other countries if we do the same things wrong, which is same herbicide year after year after year after year.
Okay. The next question is, will the cases of herbicide resistance documented in New Zealand be communicated to Ian Heap of HRAC so that they can be captured in the International Herbicide Resistance Weed Database?
So Ian Heap requires a fairly high bar for the cases to be put there. He requires evidence of a dose response curve being produced. Now that is a lot of work and quite costly. So we found the herbicide simply by testing it at a label rate, and if the chemical was controlled at the label rate, in other words, susceptible ones, and then the resistant one wasn't, we said that was resistant. We then have to take it on through a whole dose response curve experiments, either 1 or 2, to get it up to Ian Heap's standard for inclusion. We have done that for some species. We're continuing to do that, but funding is a bit of a problem for some of these plants. So yes and no, we're trying. Maybe we're too trying.
Okay. Could you explain how predictions have been made on which weeds will develop resistance?
You want all my secrets? Okay, so in the course of what we did, we looked at different things. So Phil Hume at Lincoln University did some fairly high modeling. He linked New Zealand to 11 countries in Central Europe as being the closest to New Zealand in terms of climate, agricultural practices, herbicides used, and crops. So that's one place that we should be looking at, not necessarily Australia or America, which is where we tend to look at. The other thing is another high predictor is prevalence. So the weeds that are most widely spread are likely to be the ones that evolve resistance. This is simply a numbers game. When you have 1 in a million or 1 in 10 million, the more weeds, the more likely it's to happen. And then there are what is being resistant overseas, as you just mentioned. Using Ian Heap's data is also very useful. So we have a range of tools that we have used to predict herbicide resistance. I don't know that I've covered them all, but I've given you a bit of a nutshell. You want more data? Get to me afterwards.
Cool, thanks. Are Paul Neve's findings being considered by HRAC?
As far as I know, this is so fresh off the block that I don't know yet.
Okay.
Sorry about that.
And why is it the case that weeds can only develop target site resistance to trifluridin group 3? And this was mentioned in one of your slides.
It's the nature of the molecular makeup of the plant, I would believe. I can't explain, and I don't know that we can predict any way whether a plant will do target site resistance or non-target site resistance. Looking at the mode of action, they actually, group 1 and 2 did both, group 9 did both, but then when you looked at the numbers, actually the non-target sites were grouped together and the target site ones grouped together. So that would be something that we possibly have to look at and develop more in the strategy. That's why I say this is really just an emerging concept and problem. And yeah, hang in there. We will look into it and get back to you on some of these things. We will stay engaged. I think that's the important thing. We will stay engaged one way or another with industry, with growers.
Thanks, Trevor. Got a couple more here yet. Is there new research that outlines how different post-emergent herbicides... Someone's moving the screen on me. Is there new research that outlines how different post-emergent herbicides are cross-resistant? This will be helpful when developing mixture strategies.
Once again, if it's the same mode of action, then cross-resistance is always a risk. But we need to do it on a case-by-case basis because it isn't always. So if we look at the FOPs and DIMs, for example, if you use clethodim and it becomes resistant to clethodim, it will also be cross-resistant to haloxyfop. But if you lose haloxyfop first and it becomes resistant to haloxyfop, it is not yet cross-resistant to clethodim. I have not yet seen a good explanation of that. It's just a fact that we have learned through experimentation all around the world. So experience is where we can look, and this is where Ian Heap's page is so helpful, what's happened overseas.
Okay, here's a one about terminology. What's the difference between herbicide tolerance, hard to kill, and resistant?
They're all spelt differently. So plants, because of their makeup and that some are tolerant to herbicides, it's just the nature of the plant. So a tolerant plant isn't resistant because usually you can get a rate of herbicide that will kill it. And some are specific. Some herbicides are specific to monocots, some to dicots. That's another avenue. We've just got to realize that all plants are different, basically. And that's why we have tolerant plants, plants that can't be killed. And that's the basis of crop selectivity. So we're lucky we have it. because otherwise we wouldn't have herbicides at all. So crop selectivity is based on that different nature of the plants and finding a herbicide that'll kill A but not B. And so the most difficult thing possibly is finding herbicides that kill grasses, which are monocots, and cereals, which are monocots.
Thanks, Trevor. We've got another 4 or 5 questions, but I think we're in the conscious of time, we can actually answer these offline later on. So if we move on now to our next presentation. Thanks for that, Trevor. Appreciate it.
Thank you, everybody.
Yeah, I think we're good to go. Now that Trevor's completed, we've got Rob Beresford now to give us an update on fungicide resistance management strategies.
Okay, good. Thanks very much, Paul. And thanks, Trevor. Very interesting talk on herbicides. So I'm just waiting to get control of the screen here. So when I do get control, my talk's going to be about it's the equivalent of Trevor's talk on fungicides, so fungicide resistance management strategies, and I'll cover a bit of the background about fungicide resistance and about the development of the resistance strategies and that kind of thing. And I'd just like to acknowledge Mark Christie, who was very closely involved in the updating of the fungicide strategies. So the control of diseases of fruit, vegetable and arable crops in New Zealand and worldwide has come to rely on fungicides. And without chemical protection, fungal diseases would prevent economic production in many crops. But many of the plant pathogens that cause diseases are adapting to the presence of fungicides in the environment, and this is making them harder to control. So fungicide resistance affects the most effective fungicides. Modern synthetic fungicides with modes of action that target specific biochemical pathways in fungal pathogens are called single-site inhibitors, and these are at risk from resistance. Whereas older multi-site inhibitors, which disrupt multiple biochemical functions in fungal cells, generally are not at risk. Sometimes there's other names used for these groups of fungicides. For example, single-site inhibitors are often called systemic fungicides, curative fungicides, whereas the older multi-sites are often called broad-spectrum fungicides or protectant fungicides. So crop protection fungicides as a whole are grouped by the Fungicide Resistance Action Committee, which is known as FRAC, according to their mode of action. And this parallels the herbicide situation, but the numbering systems are quite different from herbicides. All the active ingredients within a group are likely to select for resistance in the same way, which makes a pathogen cross-resistance to all members of the group. Similar to herbicides, but different mechanisms involved, obviously. So fungicide resistance has been recognised as a potentially important problem in New Zealand since the 1970s. It tends to show up in crop sectors that have high resistance, high reliance on fungicide spray programs. And the number of pathogens developing resistance is increasing steadily and at a faster rate than new fungicide chemistry is being developed. And that, of course, is a bit of a worry. Not all resistance detected in plant pathogens causes disease control problems, but certain fungal pathogens are able to acquire resistance to several single-site inhibitor groups that can reduce control efficacy. And this just shows a table of some particularly problematic pathogens in New Zealand as far as resistance development goes. Botrytis cinerea could arguably be the most problematic one and the one where there's the most cost to our industries because it affects so many different crops. I've got a list here of grapes and berry fruit, cherries and apples, but of course it also affects many vegetable crops as well. And some of the other ones here are also internationally known to develop resistance very readily. Venturia inequalis, which is the apple black spot or scab pathogen, and Monolinia fructicola, the stone fruit brown rot pathogen. These ones are also worldwide known to develop resistance readily. Powdery mildew of grapevine, a somewhat newer one, which is currently very problematic in the onion industry, is stem-phyllium leaf blight. Potato early blight is another one, and late blight, of course, potatoes as well, develops resistance to some fungicides. And then there's also arable crops and the wheat and barley and so on, where there's a number of very important pathogens which can develop resistance to fungicides. So just going through some of the important frac groups that are affected by resistance, the first one here are the MBCs, the old methyl benzimidazole carbamates. They were first used in New Zealand in the 1960s, and the resistance risk classification by frac is for high risk. That whole group of fungicides is hardly used now, and complete resistance is common. So and resistance actually started to be detected in that group in the late 60s and 1970s. So, but they're still used in some crops for some purposes. And the dicarboxamides, another one of the older groups, medium to high resistance risk is classified. For the dicarboxamides, there's a lot less use than there used to be, but resistance can be managed in these to some extent by limiting the number of applications. The demethylation inhibitors is the most widely used group of fungicides across disease control in crops. And we're seeing with the DMIs a concerning loss or erosion of efficacy across many pathogens. So these have been in use since the 1970s, and gradually there's new active ingredients being developed and released, and the newer ones have always worked better than the older ones, which have often succumbed to resistance. But the whole group as a whole, we find that the degree of resistance within these main plant pathogens is gradually increasing over time. The phenylamides is another fairly old group, active against oomycetes like the Phytophthora and downy mildews, and resistance develops quite readily to that particular group. The SDHIs is an important one as well, because that was really seen as the replacement for the DMIs as they were losing efficacy. They were released in the early 2000s, the second generation SDHIs, because there were SDHIs going back to the 1970s again, but they developed new chemistry and much more active molecules, and they were released in the early 2000s. Medium to high resistance risk, but we do find that already resistance to those is developing in a number of important pathogens. Then there's the anilinopyrimidines, the group 9s, medium resistance risk, and resistance is often detected in these ones, and a gradual erosion of efficacy in many cases as well. And then there's the quinone outside inhibitors, the QOIs, rated by FRAC as high resistance risk, released in the late 1990s, and they've really followed the same path as the MBCs, with very rapid development of resistance, and the usefulness of that whole group is greatly diminished. So a question of how can resistance be managed? The resistance development to an at-risk group can be slowed by strict management of how the group is used. And I emphasize that they can be slowed, and I think increasingly, I think we're realizing that you can't actually prevent resistance if you're using an at-risk group and you keep on using it against the same pathogen. Eventually resistance will develop. It's just a question of how quickly. So the main thing that you can do is to limit the number of applications. So there's no real magic bullets to slowing resistance development. You can just limit the number of times they're used. You can mix at-risk fungicides with fungicides not at risk. With the plant pathogens and fungicide resistance, a little bit of a debate similar to what Trevor was describing for herbicides, about whether mixing slows resistance, and particularly this one of alternating at-risk fungicides with fungicides in different mode of action groups, whether you can, well, we know that those methods slow down resistance, but resistance can be controlled in different ways genetically, and whether some of the ways that resistance is controlled is more or less increased by mixing and alternating. And then very important also for resistance management is optimizing spray application with well-calibrated equipment to ensure uniform coverage and use of disease forecasting models for optimum spray timing. So those are not to do with the fungicides directly, but they're really important. Spray application technology is crucial for resistance management, because if you have uneven deposition of fungicides in a crop canopy, the pathogen is exposed to doses which might vary from being lethal to sublethal, and if you've got sublethal dose, then you're very likely to select resistant strains of the pathogen. And also, non-chemical disease management practices are important in resistance management to take the pressure off fungicides. So crop hygiene to prevent pathogen carryover, crop canopy management to make the environments less suitable for disease, and also biocontrols perhaps have potential to play a role in resistance management, but generally their efficacy is too low to be useful as mixing or alternation partners for single-site inhibitors at the moment. We're hoping that might change in the future, but at the moment, we don't recommend including biocontrols as mixing partners. Now, there are some not so good resistance management strategies, and it's concerning that some products on the market are mixtures of at-risk single-site inhibitors in different mode of action groups. For example, groups 3, the DMIs, and group 7, the SDHIs. We see some of this, and there's been a lot of this seen overseas as well. It carries risks, because if you have two at-risk fungicides together, it means that resistance to one may develop unnoticed, which will accelerate resistance development to the other, because then the other becomes effectively a solo product, and lead to early loss of efficacy in both groups. And there are examples where this has happened in New Zealand, it's a bit of a problematic one, because it's an important marketing strategy for the chemical manufacturers and chemical sellers to have a diversity of products in their portfolios. But really, well, there's a point, I'll just go to the last point first, that mixing is best done with a multi-site inhibitor and at an effective dose, although this can be difficult to achieve in some crop spray programs. So there's a lot of complexity around designing spray programs to maximize resistance management. And you must remember always that it's very difficult to identify which component of a spray program is failing when the spray program contains multiple fungicide groups all applied at different times. So fungicide resistance management strategies in New Zealand. These were first developed in the late 1980s by the New Zealand Committee on Pesticide Resistance, NZCPR, which is part of the now New Zealand Plant Protection Society. This is just an image of that first paper, which describes the formation of the New Zealand Committee on Pesticide Resistance, and then you'll see that it's, of course, the New Zealand Weed and Pest Control Conference, or the Weed and Pest Control Society, as NZPPS was then called. So NZCPR set up the task groups covering resistance management for fungicides, herbicides, and insecticides. And the strategies are maintained on the NZPPS website, as Paul has already said to us. But another role that NZCPR had was in conjunction with AgCarm, which is now Animal and Plant Health New Zealand, introduced the International System of Product Labelling into New Zealand in 2009. And Trevor has touched on this for herbicides. And the key thing about this is that product labels, and this is just an example of one at random, cereal fungicide. The group name, the group number is put very clearly on the label, so that the users of the fungicides can see at a glance exactly which group they're putting onto their crop. so that they can help them to avoid successive applications of the same group and that kind of thing. And of course, if there's more than one active ingredient in the product, then it'll show both of them, or however many there are, on the label. And then the other aspect of labelling has been the resistance management statement, which should appear on all labels, although it's quite variable actually how this has actually been deployed. So this just describes what the fungicide is and what type it is, the warning that the resistance could develop, and there may be some information there about how many applications of that particular fungicide should be made in certain crops. Although, of course, a small label like this can't contain all the detail about all the uses of that particular product. So as Paul had mentioned at the beginning, NZCPR is a voluntary group consisting of chemical industry representatives and government scientists that developed and updated strategies until about the 2010s. But with the changing science and commercial landscape and the increasing number of resistance cases, the demands of developing new strategies and updating old ones has become impossible for the NZCPR. And so the Lighter Touch program, the ALT, which has been able to step in and actually fill quite an important void in terms of updating the strategies goes. So the fungicide updates are mostly done, and included in them are updated lists of available fungicides in different mode of action groups, simplified information and removal of out-of-date information from the strategies, and harmonised information between the mode of action group and crop strategies. And there's also some new crop strategies have been developed, particularly by Mark Christie. But it's important to note here that the mode of action group maximum numbers of applications and mixing alternation recommendations are mostly unchanged at the moment as a result of this review, because this requires new resistance data and detailed consultation with crop sectors and the agrochemical industry, and there hasn't been time to do that for all the strategies that we have. So we've got 13 mode of action strategies currently, and these are the updated ones which are now posted on the NZPPS website. And note that they include both fungicides and antibiotic bactericides. Down the bottom of the list there, you can see the antibiotic bactericides, which are used in some horticultural crops, particularly for Pseudomonas syringae control and some other bacterial diseases as well. So I'm not going to go through all the groups, but you can see that there's a reasonable number, but there's, of course, this is only about 13 out of 50 mode of action groups that exist around the world, and not all 50 groups are represented in New Zealand, fungicides that are used. But we've developed the strategies for some of the key groups. Now, this is an example of one of the information contained in one of the group strategies. This is this succinate dehydrogenase inhibitors, group 7, and the information here, I'm not going to go through the whole thing, I'm just going to give you an outline. It starts off with a product perspective, which is like an introduction, notes on the history and the chemistry and of the mode of action and the range of activity against pathogens. So that's just general information about the group. And then it provides a table which lists all the currently available products and their active ingredients, and the chemical groups within the mode of action groups that these different fungicides belong to. And then there's another table which lists all the crops and the pathogens that this group of fungicides is used against. So this is not the list of resistant pathogens and the pathogens resistant to that group, but it lists all the uses of the group in New Zealand. And then it moves on to the detailed information about the resistance. So it gives current status of resistance in New Zealand and overseas. And then the resistance management strategy details general guidelines which apply across all crops that the fungicide group might be used in. And then for individual crops, it lists the recommendations in terms of maximum number of applications, when mixing and alternation should occur, and all those kinds of things. And then there's some information at the end of the strategy on implementation of the strategy, and this specifies the product labelling with the mode of action group and information about adherence to the resistance management guidelines, as I showed you earlier. So in addition to the mode of action groups, we've got 9 crop-based strategies as well. And these consider all the diseases affecting a crop and the interactions between fungicide groups and the different pathogens. So it's the overview for a crop of all the pathogens that are in the crop, and how different fungicide groups can be used to control those different pathogens. And so these are the ones that we've got. We've got 2 in grapes, one specifically for botrytis and one for powdery mildew, summer fruit diseases. That's quite a simple strategy. That's really just a wall chart that the summer fruit industry put together some years ago, and it probably needs some updating now. Then there's wheat septoria, barley ramularia. Those are 2 newer ones. Onion downy mildew, potato late blight, and then for the myrtaceae for the nursery industry particularly, myrtle rust resistance management, and then kiwi fruit PSA for the specifically for bactericide use. So just touching on the importance of resistance monitoring. Resistance monitoring is needed to determine the frequency of resistant strains in the population and develop evidence-based management strategies. And monitoring is quite an expensive exercise, and we haven't done as much of it as we need to do. It requires surveys of pathogen populations, so that's collecting diseased plant samples, isolating the pathogen into pure culture, and then testing using either plant growth inhibition at different fungicide doses, or molecular gene detection of the frequency of known resistance genes, or in some cases plant-based tests for infection inhibition by fungicides for biotrophic pathogens, and so that includes the rusts and powdery mildews and downy mildews, which you can't grow in agriculture. And for a lot of the resistance monitoring that we do, particularly the stuff that's growth inhibition based, like for example on agar plates, the key measurement of resistance or sensitivity, there's 2 sides of the same coin. You've got a sensitive population of pathogens at one end, and then the sensitivity decreases and resistance increases as you go through the scale. And what we find in populations in terms of the percentage of isolates that you can test for resistance, that in a highly sensitive population, the population tends to be skewed over towards the left side with a lot of highly resistant strains, and very often there's a tail of quite resistant strains. But as over time, with years and decades, depending on the fungicide group, with the repeated use of a fungicide, you find that the distribution changes, and so that you end up with more and more highly resistant strains. And at some point along that that evolution of resistance, you start to lose disease control. And there's a number of cases in New Zealand where things have got to that stage, but there's a much larger number of cases where things are perhaps still towards the sensitive end, but over time are trending towards the more resistant end. So the EC50 is the fungicide dose that inhibits 50% of the population, and it's measured in milligrams per litre or parts per million, same thing. So with the increasing rate of discovery of new fungicide resistance cases, New Zealand does not currently have the capacity for all the surveying and testing that will be required over the next 10 years. And that, that's just in order for us to keep on top of how resistance is evolving and the threat that it poses to a lot of crop protection. So Plant and Food Research has actually initiated a project, and this is an internally funded project, to increase capacity for rapid pathogen resistance detection and population analysis. And this will allow growth inhibition and gene detection methodologies to be streamlined and greatly expanded. So just a final comment on the hope for the future. We've got to try and take the pressure off these fungicides. So the first thing that we need to do to preserve the at-risk fungicides is to adhere to resistance management guidelines. This is crucial in the short to medium term to prolong the useful life the at-risk groups. But the way forward in the future has to be to reduce reliance on fungicides through new research to transition cropping systems away from agrochemical dependence and towards use of disease-resistant cultivars, cultural disease management practices, and integration of biocontrols if we possibly can. So that's the end of that. Thank you very much.
Thanks, Rob. We do have a number of questions, and we probably don't have time to manage all of them. And certainly, please keep the questions coming in, because we will attend to those offline if we need to. The first one, Rob, is what impact does repeated use of half or lower rates of fungicides have on the development of resistance, i.e., does it speed up resistance or slow it down compared to the use of label rates?
Yeah, that's the big question. That's a widely debated question, and there's been some papers published internationally on that, mostly from modelling, because it's very difficult to actually study the evolution of resistance in the field in a way where you can gather data which sort of proves one way or the other. The consensus seems to be that whether reduced rates increases or slows the rate of resistance depends on the mode of action of the fungicide on the fungus. And so the resistance can be controlled in the pathogen by single gene resistance, where a single gene can mutate and then give resistance to the pathogen. And that's often very rapid increase of resistance. And the MBCs and the QOIs are the classic cases of single gene resistance, which develops very quickly. On the other hand, resistance can be conditioned by multiple genes in the pathogen, and the DMIs would be an example of this, where resistance tends to evolve gradually as the most sensitive end of the population is killed by the fungicide when it's used, but you tend to leave that more resistant tail behind. And over time, that resistant tail can effectively evolve the pathogen population into a more resistant population. So the thinking is that reduced rates may tend to increase the rate of that directional shift in resistance, that multi-gene type control of resistance may be more likely to be accelerated by reduced rates. Whereas there are arguments that suggest that reduced rates reduce the total exposure of the pathogen to the fungicide, and that reduces the rate of resistance development, and that may apply more to single gene resistances. However, there is a ongoing debate, and I don't think anyone's got the definitive answer at this stage.
Well, thank you. This will be the last, I think, before we move on. What empirical evidence do you have that biologicals are not useful as a mixing or alternative partner for managing resistance? Recent work at Cornell shows that dose and timing was important for black spot control or resistance, but mixing partner had little to no effect.
Okay, I'm not familiar with that Cornell work. And empirical evidence, well, I've been involved in some biocontrol trials where there is some efficacy, but it's maybe no better than and possibly less than many of the relatively inactive fungicides that are used against a pathogen. So the level of efficacy, and I think it's widely recognised that efficacy of biocontrols is not huge. And it's variable. It's not reliable in the sense that they don't tend to perform too well under high disease pressure. And at this stage, I don't think anyone would be confident to recommend mixing a biocontrol with a single site inhibitor fungicide as a resistance management strategy, just as you wouldn't recommend mixing some of the less effective broad spectrum fungicides. You've got to be able to prove that you've got efficacy of the mixing partner standing alone against the pathogen, which is adequate to suppress the population of the pathogen.
Excellent. Thanks, Rob. Thanks for that. Right, interest of time, I think we'll move on now and explore the insecticide portfolio with Tim Herman.
Thanks, Paul. Okay, just going to get my remote control. Brilliant. Okay. Yeah, as I said, thanks, Paul. I appreciate the opportunity to talk to everyone, and greetings to. Just trying to see how I now move between slides. Okay, first of all, like Rob, I acknowledge the work that Mark Christie has done in revising the insecticide strategies before I've come along to help out. And I'd just like to note here, I was introduced as being from BASF, who my employer is, but today I'm talking as Tim Herman, entomologist. I'm a real bugger, and just to take any potential conflict of interest off the table, the revisions I'm doing and the time I'm spending here at the moment, I'm on my own time, and just to add to that, BASF doesn't have any insecticides registered right at the moment. So in terms of an introduction, we've sort of covered this a bit in some of the earlier presenters. The task, I call it a task force by mistake, task groups were set up in 1987 with the first resistance management strategies published. Another round of workshops were done in the 90s. Bruce Chapman at that stage became the chair of the insecticide task group. We had some new strategies published in the mid-90s, and as we've all noted, that all this was done voluntarily, but it was done in good collaboration amongst the various groups. Nick Martin, who was Crop and Food under the auspices of the Plant Protection Society in the early 2000s, coordinated another update of the resistance management strategies, and you'll see on the website at the moment, most of the resistance management strategies have a publication date of 2004, so they haven't really been changed or updated since that time. And then, the task group is basically in quiescence unless action is needed, and so every now and then it does come to a fore. A few volunteers get together and we will work on something or have worked on something. But Bruce Chapman stepped down as the chair in 2014, and the baton as the chair for the group has been passed around various people. But as I said, we're in quiescence because nothing much has been needed. Now, as part of this, because a lighter touch is managing this update, the ovine pest, the two strategies for blowflies and sheep louse have not been included in these revisions here. We noted earlier that the Insect Resistance Action Committee, it's a global group, was set up in the mid-80s, just prior to our strategies getting set up. And the way the insecticides are classified in their strategies, so they're classified on the how they act, whether they're in the guts, in the nervous system, or respiration, whatever, and then where they act, specific sites within those things was set up by IRAC, and that poster is readily available on the website. But we use that classification system here as well to keep it all uniform. We've put that website there. It's a very good site for resources on the background information on insecticide resistance. I'm not going to spend a lot of time dwelling on that here and now, because hopefully most people are well aware of that sort of information, but it's there if you need to go through to it. So the individual resistance management strategies, the first ones in the late 80s, early 90s, were in response to field control failures, particularly the organophosphates that had been used a lot, the carbamates, and then the synthetic pyrethroids that came through. So those strategies there, they were because the organophosphates, broad spectrum insecticides, had knocked out a lot of the natural enemies and so forth, they were applied on a regular calendar basis, and it was not very sustainable in the long term. We had resistance coming through. In a couple of instances, and I note one there, that tomato fruit, we noted that sensitivities, in this case it was to synthetic pyrethroids, that was declining. We found that in testing we did of the adult moth, well below a level where you'd get field control failure, but it was just the canary in the mine to say we've got an issue developing if we don't change our management. And that was some of the impetus between that led to IPM programs being implemented out in the fields. Yeah, and as I said back then, control programs were largely based on calendar spraying, but we were moving towards IPM programs as we were developing them and bringing them through. Then the later programs that were developed were due to concerns. We were seeing field control declining. Onion thrips and onions was a classic, but also there was new incursions of existing strains of existing pests. Western flower thrips was one. It was already here in lupins, but not really much of a pest. Then it was found in greenhouses, and that was proven to be a different strain of that insect, and it had resistance to a level. We also had the potato tomato psyllid arrive, a new pest to New Zealand, and we had to develop a resistance management strategy for that, or more of a insecticide rotation strategy to avoid resistance. We didn't have any documented resistance to it. But yeah, so with that TPP, the tomato potato psyllid, we had existing IPM programs in use in tomatoes and to an extent potatoes, and those just got thrown out the window when the psyllid arrived. And then once we adjusted to that new pest in the crops, we then had to bring in a programmed approach. So that's one of the more recent strategies that has been developed, so that we can rotate the chemistry, use softer chemistry, enhance or promote the natural enemies as we move through. And as I note there, the IPM programs are used a lot more. There were formal programs developed with government funding. The tomato fruit worm was one, diamondback moth was another, codling moth work was done using a degree day model to time insecticide use and so forth. And for codling moth and apples, that's a critical one because it's a quarantine pest going into some export markets. So if we don't manage it appropriately, we won't get access to those markets. So that does really put the pressure on making sure our programs are up to scratch and tight. We did have some resistance developing in codling moth to one of the newer, more selective insecticides, but we have now managed to reduce that to a level that hasn't gone to control failure. And then I note there that we've got two draft strategies that are just coming through the system, Nisia sweetbug and the tomato potato psyllid, and those are the only two really new resistance management strategies that have come in the last 20 years. So I'm not sure whether we take that as a pat on the shoulders or pat on the back that we've been doing such a good job, we don't have any resistance issues out there, or is it because we're not looking hard enough? Because I do know, unlike the herbicides and fungicides of recent years, we haven't been doing much testing of our populations out in the field. So we're only going to see something if it suddenly became resistant and we got control failures out there, which is a wee bit of a concern. So, whoops, I've jumped the slide. No, I haven't. No, that's all right. So the mode of action table within the on the NZPPS website where the resistance management strategies are hosted. That's been updated. I've introduced the colour coding to match the colour coding of the IRAC chart, so that the different areas of where they work, a group. You've still got the groups of where they, the actions, mode of actions that they have are in there. It's just to try and make things a little bit easier to follow. So that if we just go to the next slide there, one too many. Back up a page, previous. Too fast, excuse me. Got a twitchy finger. There we go. So that's a snip, should I say, out of the table. So you can see there's these three different colours of the different groups, and then within those higher level groups, we go down into the mode of action groups, and then the active ingredients out to the right. So that will be loaded up in the next week or three, hopefully, as we finish getting them all done and through the system. So then within a resistance management strategy, this is obviously leaf rollers. I've added a snip there, or a note there with a link to the ACVM website. That is theoretically where the most up-to-date lists of active ingredients and registered products are. You have to search for them individually. You can't search by pest or crop, but that is a way just to check where things are, because they are moving. And the reason I took this snip is because with the OPs, the organophosphates, there has been some changes in recent years, and there's some more changes coming. So some of the older ones have gone, and the ones that remain, such as acephate and diazonide, you have restrictions on their use, that they can only be used as per the label, and you cannot use them off label. And growers need to be very careful with that. And even within, say, for diazonide, for example, it's registered on pip fruit, but it can only be used on non-bearing pip fruit trees. So that's where some care will need to be taken moving forward. So where to from here? We've just got to complete and load up the revisions that we've been doing, and reinvigorate the task group. In the past, when we were first set up, I was a younger junior scientist, early days of my career, like Trevor. We did a lot of collaboration and communication around the CRIs, the universities, and the ag companies. And we've got to make sure we bring the growers and the grower organisations along, because we are going to rely on them. These strategies of guidelines, they're not a prescription, and we need to make sure that everyone works together to make sure it's the benefit of them all. In terms of the future, because we're moving away from I think we have more or less moved away now from all the older broad spectrum chemistry, and we're coming to the softer chemistry. We're encouraging the beneficials, and the leaf rollers are a classic where they used to have resistance, whereas now we don't need to spray them much at all, because the beneficials, the parasites out there, are keeping them under control. But we've got a real push into sustainability, regeneration, integration, and it's just being emphasised a lot more. So once we've done this workshop, sometime in the not too distant future, we need to get the funding together and get out there and do some more work, and hopefully in the insecticide space start doing a bit of assessing and testing of populations to make sure we're managing them correctly. So from there, hopefully it will be a new dawn of the resistance management era. Thank you.
Excellent. Thank you, Tim. We've had a question pop up while you were presenting, and I'm not sure whether it's specific to insecticides, but interesting to contrast resistance and its management in weeds, arthropods, and pathogens. How much crosstalk is there between scientists working on these three pest groups, and how much overarching research from an evolutionary ecology perspective? I can't answer that because I'm not a scientist. So
yeah, that's quite a high level question from Murph there. We do, to a degree, and I call in the past, and like a classic was in the apples, the use of mancozeb was found to, it's a fungicide, a broad spectrum fungicide, and that was impacting on predatory mite populations, leading to mite flares. So where there's direct linkages, definitely they're working together. But Rob and Trevor and I bump into each other at conferences and so forth. We're always having a chat. How formal those crosstalks are, it depends on how much need there is, and we do, I guess, keep an eye on what the other disciplines are doing, and if there's anything that needs to be or is overlapping, we will engage. As I said in my last slide there, communication, collaboration is important.
Yeah, cool, thanks. I might be able to answer this. Next one is, how many views does the New Zealand Plant Protection Society website get? At the last AGM, Mike Barley, who manages the website for the Plant Protection Society, did put up some figures, and I can't recall what they were, but I'll report what they were. But the other thing too is they're able to identify where the traffic comes from, and I think a lot of it is from overseas. A question here, which is probably a general question. If the same active ingredient is used as a seed treatment, nematicide and fungicide, does this increase resistance risk, or is it minimal due to the different targets?
My comment on that would be it needs to be factored into both areas, into the the pathogen that the fungicide is targeting, and the nematicide, the nematode that the nematicide's targeting. So it sort of bridges into the two different resistance management strategies. You can't ignore it; it needs to be considered, and that's also where there's potential. If you use an insecticide for a caterpillar, it might also have an impact on an aphid. So you've got to make sure it's picked up on both resistance management strategies if there are ones. You don't just look at the products that are targeting the pest in the broader sense of that resistance management strategy. I don't know if Trevor or Rob want to say anything on that.
I could make a comment too. For the fungicides and plant pathogens, the seed treatment one is one very good question, and I don't think there's a definitive answer. Another one which is similar to that is the one about for example, for a plant pathogen or plant disease, winter use of a fungicide to control it in the off season, and whether that should count towards the in-season use of the fungicide in terms of the numbers of counts for the limitation on maximum number. There's no empirical evidence available for either of those questions, but prudence would suggest that you're potentially exposing the pathogen to this fungicide in this situation, either on seed treatment or in the off season. Therefore, there's a risk for resistance selection, so that those uses should also be built into the resistance management strategy.
Well, thanks, guys. It's another question which relates to fungicides. Overseas, does the rate of resistance correspond to the introduction of fungicides with new modes of action and alternation of fungicides in arable crops?
So the question is, for arable crops, what is it, the rate of introduction?
Does the rate of resistance correspond to the introduction of fungicides with new modes of action?
The rate of resistance, well, it depends on the mode of action, and certainly with the single site inhibitors, and particularly those ones that have got single gene control of resistance, there's very rapid resistance development and when the new fungicide's introduced. And unfortunately, there's a tendency for a new fungicide group, when it's introduced, for it to become the flavour of the month or the year or the decade, and for them often to be overused. The QOIs, again, is a classic case, particularly with cereals in Europe, where the QOIs actually cause leaf greening, and you can actually get a yield enhancement without the pathogen even being present by using the fungicide. And that was one of the contributing things that led to the very rapid demise of the QOIs in our crops.
This is a general question. Should New Zealand strategies always adhere to FRAC, IRAC and HRAC guidelines in the first instance and adjustments made as data comes to hand locally?
Certainly for fungicides, that tends to be the way it's happened. And that's the important point, is that we need to start somewhere, but we also need local information as to how we should really be managing them in New Zealand.
Yeah, I would agree with that, Rob. You know, the scientists involved in the IRAC, FRAC, and HRAC groups know the chemistry a lot better than we do. So we should really adhere to their recommendations to a large degree, and then if we tweak it as we need to with local data, but also feed that back up into those groups so that they're considered and get feedback on that as well.
I would add to that and just say that the size of the research organisation and base in New Zealand makes it essential that we use a lot of this overseas information and capture as much of it as we can.
Here's how do we promote the use of resistance management strategies for wider adoption? Are there success stories here or overseas we can use as good examples?
I think in the case of fungicide resistance, a lot of the action, the implementation of the strategies come through the sector associations, the crop sectors, and the crop sectors have varying degrees of control over their members. But for the big export crops, they have a lot of control over what their members can do because of the critical aspect of fungicide residues on the crop, and so usage of spray programs is fairly tightly controlled, and therefore resistance management can be very tightly controlled. And I think increasingly for fungicides, the crop sector associations are really taking the lead more on the development of the strategies.
Yeah, I'd agree with that. You know, there were examples in the early days where programs were developed but not implemented because there wasn't a driver. But if there was a processor or a marketer on the other side that said you must use it, then the growers were pushed into it, or the strategy sat on the shelf until they started to get resistance, and then, oh, we need to use this, and they were pushed into it. But it is a good question that we do, and that's where we need to have the buy-in from the growers. And I think they're a lot more switched on that they need to minimise their impact out there in the environment, and only spray when they need to. And you know, they're picking up, they're seeing the benefits of doing better crop monitoring and assessing what's out there before they spray. I think calendar spraying is, to a large degree, no longer used unless it's part of, like you do have to do it to a degree for fungicides, but for a lot of other things you can manage it more carefully.
I think another comment would be that resistance management for any of the pesticide groups, it's a three-way thing. It's the science, it's the agrochemical companies and the suppliers of the chemicals, and it's the users and the crop. So there's the crop sectors, and so it's a dialogue that has to happen in between those three groups, and without full consultation, it's very unlikely the strategies will be taken up, because if the agrochemical industry doesn't agree with what the science is saying in terms of the way it's converted into strategies, well, then things won't progress, and if any one part of that triangle is missing, it's just not going to work.
Do we have New Zealand representation on FRAC, HRAC or IRAC?
No.
Yeah, my impression is that given that the groups that we had in place have been almost disbanded, and there's never been a person that's been targeted as being responsible. But for FRAC at the moment, I've continual communication in email, and when the I think it's the Southeast Asian group have meetings I've attended. So and somehow by default I've managed to be the New Zealand representative. So I'm not sure how that happened.
Well, my comment to add to that, Paul, is that the agri-chemical companies have linkages up there, and that a lot of the scientists who've developed these modes of action chemicals are representatives on there. So internally we can link into them. Yes.
Yeah, no, absolutely. I'm aware of that with FRAC as well. So yep.
Yeah.
One for you here, Rob. Thanks for your presentation. In reference to your closing remark, what policy, societal or other changes need to occur to convince growers to implement more resistant cultivars in their programs to reduce reliance on crop protection compounds, but not be penalised for the likely reduction in yield outcomes?
That's a big question. It's a very important question. It's frustrating in many, well, for a plant protection person, it's really frustrating that we see these susceptible cultivars continuing to be grown all the time, and they can only be grown because there's pesticides to prop them up. So I don't know the answer to it actually. I think that the real trend will come through a crisis. So we'll probably start adopting resistant cultivars, and there's resistance out there for many crops available, but often it's not associated with the other more important agronomic or marketing properties that the plant breeders tend to prioritise. So with the pesticide resistance crisis, which I believe is looming, I think it's only going in one direction. That's getting more difficult to control pests with pesticides. I think that will tend to force the hand, and that hopefully we'll see more adoption of more resistance in crop cultivars.
But it's also going to be that the cultivar chosen to a large degree is what the market wants, in terms of flavours and size, colour, whatever. Any resistance to a pest or disease or what comes in as an extra on there. So if you do have that resistant cultivar, it's still got to be able to meet the market requirements, because you won't sell it otherwise.
This one here is about ratios. What is the established field dose to EC50 ratio threshold in which resistance is deemed present in a population?
Dose to EC50 ratio, there is no established threshold. The dose to EC50 ratio is a recent invention of mine as an indicator of hypothetically when increasing EC50, which means increasing resistance in the pathogen population, may start to impact on disease control. And there's a range of values, and at the high EC50 end, you end up with the pathogen being so insensitive or so resistant to the fungicide that it's as resistant as the field application rate almost. So there's no hope that the fungicide will control the pathogen. But the point at which it reaches that point, we don't really know. There's no definitive answer to the question, I don't think.
And can I just add there, Paul, that I was going to make this comment with reference to another question. A lot of this resistance management science is theoretical. We don't know. We can't test it and model it. Or we try and model it, but we can't do it out in the field. We don't have real live situations, and each one's going to change for a different pest pathogen and the crop they're on and the climate they're in. So it's very hard to have any one rule of thumb that this, if you do this, this is what will happen. It's quite a difficult area because you're just sort of trying to anticipate, if I do this, this will be the outcome, and you can't really do small scale scientific trials to prove it. It's very difficult.
There's another one here about fungicides. When limiting fungicide applications per season to avoid resistance, should we approach this per crop cycle or as a whole production system? For example, we could spray once or twice per chemical group per crop cycle, but different crops throughout the farm may repeat this practice, and at the end you use the same chemical group constantly.
Yeah, I get the question, and of course the strategies and the either the mode of action strategies or the crop-based strategies focus on a crop and a pathogen often, although with the crop strategy we try to integrate cross pathogens. But yes, there's no guarantee that well, you do have to take into account the entire picture of a crop. Like, for example, for apples, powdery mildew and black spot are both controlled by the same fungicides to some extent. And for example, the QOI fungicides, which are no longer working against black spot, still are effective against the powdery mildew. So there's still room in this program to have QOIs used, but you don't expect them to control the black spot. So the integration is difficult and challenging for a person trying to design a spray program. And if you've got annual crops where you might have different, if you're rotating and you might have the fungicides used in one crop and then the same ones are used again in the next crop within a year or so, yeah, it's a difficulty. And at the moment we aren't in a position to do that between crop management just because we haven't looked at that level of detail.
The other issue is that with regards to insecticides, you've got the pests don't recognise property boundaries. So you, particularly with annual crops, you get a mosaic of insecticide use across a region. And they're like, for example, with diamondback moth, there isn't an attempt to have two windows of insecticide uses so that you minimize the exposure of a group into a window so that you don't have it present throughout the season on multiple crops. And that might need to be done more and more into the future. But like apples with black spot, you've got a set window of when these products are being used, so that sort of reduces it. Yeah, but it is a good question. It is something we need to keep considering as we move forward with our strategies.
This one, I'm just going to some of the herbicide stuff, Trevor. In New Zealand, we have several non-chemical IWM tactics in action, such as crop rotation, higher seeding rates, cutting of hay, silage and grazing outside of adopting no-till cropping for winter crops, which supports new mode of action pre-emergent herbicides. What other non-chemical tactics should we be looking at?
Yeah, quite a long list there of stuff that we can do. I don't know that I can add to that at the moment. There's a whole lot of new technology coming in terms of electrical weeding, laser weeding, precision ag, cultural weeding. Who knows which of these are really going to get the broad acre and be affordable, but there's several things out there. Simply whatever technique reduces the number of weeds is the best technique to go for. And growers and farmers will have the best idea of that. In pastoral, it's reduced stocking. In arable, it's trying to keep the ground covered for as much as possible. In other crops, there's probably other strategies, but it's a numbers game. The fewer weeds we have, the less chance of herbicide resistance evolving.
This is hi Rob. Regarding your comment on the prevalence of Group 3 and Group 7 mode of action fungicides, what would be the most effective alternative approach?
It's a big question. It's a very difficult question at the current time because we're so dependent on fungicides in many crops, and it has turned out that the DMIs have been the most used, and then it looked as though the STHIs were going to sort of step in, but they're starting to fall over a bit as well. So really, this is the bind that we're in. We have no easy solutions to that, and we have to look more widely at other groups. I think we have to look at possibility of other broad spectrum fungicides, other multi-site inhibitors, which may be able to be brought into crop programs. But then you run into the problem that if products aren't registered in crops, so if there's no registration in a crop, then they shouldn't be used. You can have some off-label use, but that's has limitations. You can't just do that regardless of everything else. So it's a real bind. We're in a difficult position, especially in relation to the DMIs and STHIs, I think, because they're so widely dependent upon. So sorry, I can't answer the question better than that.
Okay, thank you. I suppose this is a general rather than specific. What role do chemical groups within a mode of action have in managing cross resistance?
Yeah, for the fungicides, it's up to FRAC. They classify the whole group as with a resistance risk low to high. They never, as I can think of, consider the chemical groupings within the mode of action group. But we do know that different active ingredients within a mode of action group do tend to develop resistance differently often. The DMIs is a classic case, but you also see it in the STHIs as well. So there is variation, and everyone recognises the variation in cross resistance within a group. But nevertheless, I think in the case of the DMIs, this is where we see this gradual shifting of sensitivity towards more resistance, even though there's new active ingredients being introduced, and for a while they look better, and they are better than the old ones which are no longer used. But nevertheless, the whole group is marching towards resistance at a rate. So I think that like 20 years ago, you'd look at the DMIs and you think, oh, there's a lot of the cross resistance within the group is highly variable, and it's not as great as you would think from the resistance rating that FRAC gives it. But now we realise that in actual fact the whole group is tending in the same direction, even though there's still variation between individual active ingredients.
I'm going to have one last question here that I think I might be able to answer. Have the ag chem companies that develop the active ingredients been consulted regarding updated strategies? They generally invest heavily in resistance management science and would likely have highly relevant data to support us in New Zealand, given the local committee is struggling to keep up to date. My understanding is that when Mark Christie did the first updates, that he consulted with ag chem companies and with sector organisations and with scientists to bring those strategies up to date. If there's a feeling that that isn't at a level that everyone would like, then this really needs to be addressed then at the workshop, in which case all registrants or AgChem company representation will be attending. What I'd like to do now is just, I suppose, sum this up, and I'd just like to say a big thank you for everybody that's attending. I think we got up to about 80 or 90 online. A few have dropped off since. A huge thank you to Trevor, Rob, and Tim for pulling together their presentations for this webinar. To look forward, I'm happy to facilitate any questions or comments that come to mind after we've finished the webinar. And there'll be a recording which will be up on the Lighter Touch website, which should be available in a couple of days. And obviously my email address is there, and I'm happy to facilitate questions and pass them to the respective panelists. Any questions that we haven't been able to answer in the time we've had here will be answered in writing, so we can sort through that. And I'm looking forward to arranging and facilitating this next workshop, where a number of you people that are online now will be attending, and will be invited to contribute to making a New Zealand pesticide resistance management strategies current and kept current. So thank you all very much for attending. And I'd just like to ask the panelists that are online at the moment just to stay online for a couple of minutes while we catch up with the rest of our Lighter Touch team. And thank you to Gina and Sarah, who are behind the scenes. We can't see them, but they've been managing the IT for us and making things work seamlessly. So thank you very much, everybody, and enjoy your lunch and the rest of your day.